السلام عليكم ورحمة الله وبركاته. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. ورب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. وبعد, brothers and sisters in Islam, Assalamu alaikum wa rahmatullahi wa barakatuh. Inshallah ta'ala, we're at our 30th, our golden seminar from UJ. May Allah subhanahu wa ta'ala, or from our golden age, may Allah subhanahu wa ta'ala accept it. May Allah subhanahu wa ta'ala forgive me for my shortcomings and allow us to reach and stay steadfast upon the Siratul Mustaqim on top up till and beyond Yawm al-Din. Ameen ya Rabb al-Alameen. Um, Ameen ya Rabb al-Alameen. So, inshallah ta'ala, students will actually be at their last for BIOA02. Class 1 will be at their last lab, week 6, inshallah ta'ala, before review, from prep for their, so lab 6 being population sampling and revisiting the effects of road salt on Lemna as we set up last week or 2 weeks ago for them, bidnillahi ta'ala. So let's have a look. We'll see what their results are. They'll come here as well to input the data, bidnillahi ta'ala. Alameen. This course is, and I wanted to actually have a reflection seminar. Bidnillahi ta'ala, we'll have this whenever Allah's most pleased. But really, being able to appreciate, Allah subhanahu wa ta'ala's creation indeed is something that, He, Ahsanul Khaliqeen, the best of all creators, has, willed us to be able to explore or to appreciate. And may Allah subhanahu wa ta'ala allow us to be sincere and steadfast upon this deen, sincerely for His sake. Ameen ya Rabbal Alameen. May He Jalla Jalaluhu fi ula allow this to be the best seminar, or may Allah subhanahu wa ta'ala allow us to be best pleasing to Him in every moment of our lives. Ameen ya Rabbal Alameen. Without any hindrance, without any interruption, without any hesitation. For indeed He is Lord of all creation. وَرَبُّ الْعَالَمِينَ. يا رب العالمين.
finally, to find the answer. What is the program here? One candidate, as well as the feedback survey. That's something we're doing, or at least other from the other company. Hopefully, that's the approach that let me know what you think about them. And again, I'm on this. You can also want to, and then one of the times you are done, you can begin the P-lab quiz. You can also begin the P-lab quiz with the quiz. And then the one that afterwards, something are
Time reminder post as you're doing your timed independent pre-lab. Bear in mind that you can use your lab manual. Even if you had a separate lab notebook, you could do so as well, but bear in mind that it's independent. So just keep that in mind. Oh, if okay. Did you see anyone outside? Anyone else from our lab that you saw coming in? Yeah. No? Okay. So in the meantime, you can wash your hands. You can get ready and then begin with the lab quiz. When you're done, and when everyone else is done, they can also do the feedback survey just before we begin the practice. So feel free to get started. Um, Can I have an update as to how we're doing for the lab quiz? How many of you are almost done? Don't feel free to take your time. Do we need another 5-ish minutes? Folks, do we need about 5 minutes? Yeah? Okay, 5 minutes. If you have questions, let me know, but don't remember this is the individual quiz, so please make sure that you're not interacting with one another. If you have questions, feel free to ask. No problem. Before we begin the lab, feel free to grab a drink of water if you need to, not fasting. If you have like gum, for example, my recommendation is to actually dispose it outside because technically you may be distracted and you don't want to have food inside the lab accordingly. So just uh, in the meantime, um, actually, I'll wait a few more minutes for those that are in midst of completing the pre-lab. And if there isn't time, then we could talk about the lab survey feedback reflections from you all near the end as well. Okay, so how are we doing, folks? Are we done the pre-lab? Folks, please make sure that we're not discussing, as some of you may already be doing the pre-lab in the midst of the chat. So we don't want people to also be distracted during that period in time. So congrats, folks. We're in our final week, our final stretch of the lab. Just to confirm, everyone has completed the pre-lab. If anyone's waiting, do you need some time? Mm, We can ask the lab tech just in case there's a possibility for him to extend it. If what you could do is go down that side, just knock on the door and ask if it's possible that you can get an extension. If that's something that you want to consider. Okay. That's okay. That's okay. Yeah. In the meantime, has everyone gone to also complete the pre-lab? If anyone hasn't, just please let me know. I have as well with us today 17 people. So we may have a group of 3 because we'll be doing exercise 4 in duos or pairs. So we'll try to accommodate accordingly. And that refers to actually our previous week's material, delivering the lemnas for exercise 4. So I'll talk about this now, but just to confirm, everyone is done, right? To feel out, you guys are done. Please, once again, folks, will pay attention till the very end, because there's much to do. But before that, once again, if you haven't done the pre-lab feedback survey, please do so. It'd be really beneficial for myself, and also to get an idea as to what I could do to improve for future, like even just in general. Um, we've done the pre-lab, lab survey. Hopefully, you've already done that as well. If you haven't scanned it, you can do so. But as you can see, we have an intro. We're going to go back to the past with Lab 5 material, talk about the lemnas. And then we're going to go to mark-recapture, which is going to be with the beans and rice that you have on your veg tops. So, oh. Yeah, so pretty much we're just going through the outline. But inshallah, the goal is we're gonna try to go into the past, complete lab 5 material, just like the winter wheat as we did from lab 1, and then continuing on with analysis in lab 2. And I think actually that's where we're gonna go straight to. Let me see. Someone else also outside? No. Oh, okay. Okay. There was something I wanted to also share. Okay, I'll try to share this afterwards. Let's go down and into road salt. Okay, so talking a bit about the bioassay, as you recall, we had different concentrations of NaCl. Can anyone just mention one of them if they recall, either from the group or from the manual? Just mention a concentration or percentage of NaCl. 5 different types. You recall? You recall the concentration? What's up from last week? I will give you a hint. Oh, yes. 4.5? 12.5, yes. That was the stock. 0.625. And that's the first experimental treatment. Dorlin, did you want to say as well? 1.25. So pretty much it doubles from 0.625 to 1.25 to 2.5 to 5. And essentially, we have the duplicates reiterating to us the effect of table salt on these lemnas. These are duckweeds, and so they're aquatic plants. And so just like how we were able to do an assay analyzing the growth of winter wheat from weeks 1 to 2, we're now in our last set of labs seeing another dimension of assays now under the sea, or in this case, in water. So really, the idea is that we would be able to hopefully extend this analysis, especially during the winter season, right? Because a lot of road salt is deposited into highways, into roads, etc., and crosswalks. And so these aquatic plants, even those that are more terrestrial, would be able to thereafter absorb or essentially be exposed to higher concentrations of these salts. So you want to think about the consequences of these results as we look at them today. These are some articles that reflect this also in terms of their impacts on fresh water. And if you recall from the zoo, actually, we talked about certain deposits that have impacts onto different aquatic organisms, right? You recall the Great Barrier Reef, that has amelia, like many types of aquatic organisms, crustaceans, fishes, etc., and plants included. So you can imagine the effects that they have accordingly on, the creation. So bear that in mind, for our nature. So we'll skip that slide because I'll talk about it. Well, actually, so I'm gonna ask essentially, um, let me go back. Yes. So labs exercise 3, you'll be able to do after the lab. If you want to stay in the lab, that's okay. But primarily we'll focus on exercise 4 for the post lab of last week. So your goal is pretty much to grab your cups. Remember the 3 cups you had per group. Remember your condition. If you're a control, if you're one of the other experimental treatment conditions, and accordingly, you would be able to look now, 14 days or 2 weeks later, as to the number of thalia that your plants have. So what I want you to do is actually grab your group's reps for the lemnas. I'm going to draw some things out, but a member of each group can be able to grab them down where it says Wednesday, 10 to 1. So right behind Owen. Uh, so if you guys want to have a look, just grab. You'll see like you're writing, so it should be familiar. And that way I'll talk a bit more. You can wear gloves. If you want to wear gloves, you can after you grab the cups, or even now. Um, Great questions. Okay. Has every group grabbed their reps? I see a few there. Remember, you would have had 3 cups. Make sure that you have 3 cups. Your rep A, B, and C for your specific condition from 2 weeks ago. Yeah. If your group was also different from 2 weeks' time, it's okay if you coordinate. It's going to make sure that as easy. As long as every group has grabbed their cups, right? I'll explain it in a moment. So feel free to make yourselves comfortable or like now pay attention actually. Sorry, with regards to what's next. Uh so your goal, and I want you to be, cognizant of this. Each bench should have three separate plates for each rep. Folks, let's pay attention. I want to make sure everyone's paying attention. It'll help it also make it easier for everyone else in your group. As a reminder, you should have maybe 2 to 3 people per group. I know some of us from our previous week, we may have grouped together. But if there's a need of help, let me know. Um, because okay, what we're gonna have or what we're going to do is essentially count the number of thalias. Remember the miniature leaves on the lemnas? So your goal is to count. Pretty much what I showed there is that you see 7, right? And not like you're not counting the plants per se, but you're counting the number of thalia, as that reflects the population's growth more reliably. Because there's variance, right? You can have a lemna with 5 different thalia or thalli versus one that has only 2. So you want to be able to see across a wide variety of those in each cup the results accordingly. Now, each person in your group can deal with a specific plate. I want you to bear in mind that as you count, try to be organized. So what I mean is that if you're counting with the loops, bear in mind that, for example, let's say I have thalia here. What I would do is, as I take them out, I look at one thalia or take a number, maybe only 2 at a time. Ideally just one, and then count the number of thalia, place it out, arrange it on the grid, arrange it on your plate as if it was a grid, so maybe on the top left or top right side, and then keep going to arrange the other ones accordingly. It'll make it easier for you, so that way, in case you have to look back, you already know the number, and you keep accumulating the data. So for example, I count the thalia, and or I grab a lemna plant, and I find that it has 3 thalli. So I say 3. Then I essentially dab it out. I grab another from the same cup. Remember, don't switch between cups, but make sure that you use each plate for a specific route to be consistent. And thereafter, or to maintain consistency. And thereafter, if I grab another thalia or another lemna, and I find 4 thalia or thalli, then you're going to take 7, right? Because 3 plus 4 equals 7. So just imagine you're counting bills, like 5, 10, 15, 20, or $5 bills. You just keep adding, keep adding, keep adding, and that will make it much more easier for you, rather than essentially using a counter or essentially doing it in another classroom. Does that make sense, by the way? As you count, once your group is done, you actually will be uploading it here onto the Excel sheet. And so, inshallah, you're going to be able to put it right here in column D, where it says day 14, exhibiting your second population. And so that way, you should be able to compare N2, i.e. your second population or your population at 14 days versus at 0 days. And thereafter, these other columns you'll be able to fill in. And this will be beneficial for your final lab assignment. Any questions, by the way? No questions? Okay. Yes, and on Quirkus, you'll be able to access this, so don't worry. That's where the other slide comes in. But the goal is you'll include this data for exercise 3 rather, and then it should be uploaded um, or it should be available on a separate sheet for Quirkus, for exercise 4 on Quirkus. Any questions, by the way? Final questions before you begin? Question? No? No questions? No questions? No questions? It's a great question. So I would recommend if you all get interested, if you find like a miniature bud, and this is actually what you often see, this is what you would see in theory, but what you kind of see is sometimes something like that, right? For a thalli, for Lemna and of this shape. I would recommend if it's relatively, even though it's small, if you see a bud that's visible, count it. So that way we all be consistent. And actually, generally speaking, it is a growing phalanx. So you can't include it. So this one would technically have 4. Right? So that'd be 1, 2, 3, and 4. And so you just want to keep counting with your group. Try to do this in the next 20 minutes because each person should be able to take out 1 for each rep and be able to count accordingly. And then that way we should, inshallah, be done by 11. And then you'll have more time to complete your final exercise of AO2. You're taking out all of them. So you'll have 20 minutes. But you have to take out all of them. But that's why, so for context, each cup can be done by 1 person, which is why I'll make it easier for you guys to do. If you guys need help, let me know. I would recommend if there is a group with 4 people to help out a group that only has 2 because they have 3 cups likely. So just bear that in mind if you can accordingly support other groups. If you are at a cup, please try to be mindful of time so that way, and also reliable in your counting, so that way you don't run the risk of deterring your group members. Try to take out 1 at a time. Make sure, folks, you also record the rep at the very end. That's Is that fun? Yeah.
Yeah. Okay, thank you.
No problem.
So all of them, like, take off the map here and count them.
I recommend counting them as it's
Yeah.
it's usually like 5, 9, because it'd be 5 plus
Yeah.
4.
So you take all of them and count them.
If any group is using a counter, other members in your, next copy. Try to count them, folks, as you take them out. Don't count them just on the plate. It'll make it easier to count them as you look at them. So as you take out one with the inoculating group, you'll see, okay, maybe I have 4 thalli. Then you go down for your initial count as 4. Then you find another one that also has 4. Then you total of 8. So you keep counting. They'll go from, like, let's say 4 to 8 to 11, 13. 17, 22, etc. So bring those details in mind and make it easier for counting. If your group is done, you can also move to help other groups complete theirs if they're having difficulty completing it.
How are we doing so far? Are we able to grab a number of lemnas? Actually, I'll have a look. Everything okay, folks? We have about 10-ish minutes. Let me know if that's okay for timing. If we need a bit more time, that's also okay, but just let me know. if your group is done or a member of your group is done, I highly recommend asking around if other groups need help because, as you can imagine, they may have other conditions that they may need help, your help in. So bear that in mind. I don't want to see any of you on your phones, especially when there could be people helped out by counting the remaining number of lemnas. So try to support one another because at the end of the day, we are supporting each other as a group, as a lab. Very important indeed. So please make sure that you're being mindful, aware of how you can best support one another. Thanks. Are all done, folks? You could let me know and we could accordingly input them onto Excel. Please make sure that if your group is done, they could tally the information, let me know, and other group members could accordingly help other groups because they may need your help accordingly. Have an extra? Things I don't want us to lose track of them. So you already counted these, right? Can we input these? You recall the condition in the before I do actually. Okay. Okay. Can I get your flow? 222 for number C. The control. E as in 10. Did you specify the concentration? And then rep C, right? Okay, I appreciate it. Also, if your group had from the beginning more than 12 thalia, because I know some of you may have gotten more than 12 in the past. If you recall, we had 4 different plants or 4 lemnas with 3 leaves. If you feel like you had more, it adds up, let's say to 13 or 14, let me know. So that way we can adjust to the starting values accordingly. But don't worry, you don't even have to input your data now. What's more important is that you have all groups complete accurately their counting. And that would be really appreciated.
Thank you. Well, you know, the goal or what you actually want to jump, numbers to take it easier. And this is for everyone, by the way. Place it into this hemisphere, the numbers are gonna go down. You don't want them to go down, into aquatic lungs. But essentially, let them essentially go on the plates, rinse with the water, on water that is not being absorbed into the basin, and then you should be able to, favorite.
Also, please, I do record this, so if you're not comfortable with being shown, okay. Remind me which one's yours. Experiment for 2.5 or 5? And rep 1, do you have all of them or just one? Perfect, okay. Whenever you're ready. 410, 410, okay. 53, 300, 350, 315. Okay, perfect. I appreciate it. Okay.
Yeah, the numbers that I can find the spreadsheet that is so do you know where to find it?
I can help you guys find it. I'll help you guys find it.
So, thank you.
So,
Do we enter it all together on one sheet?
yeah.
Oh,
Actually, on this sheet, and then you guys should be able to see it across the two commissions.
sure. So three of us, this is sample one. Sample A is 222. Sample B is 226.
Is it really the case that these two are the same?
Yeah, I counted twice.
Okay, okay,
Yeah, and I think C probably also recounted too.
okay, okay. I've got them all.
Okay.
I appreciate it. No, no. If once again you had an initial thalia count of more than 12 for each rep, please let me know and we can make those adjustments now. What I mean by initial is that on day 0, some of you may have gotten more than 3 leaves on each thalia of the 4 lemnas that you had. So just bear that in mind. And we can make those adjustments here because they make a grave impact in your total growth rate. So when you're done with the pond water, you can use actually the water to rinse off the lemnas off the plates. That's my recommendation. Or use the water to pour down. Not that one. Yeah, that one will make it easier because they've already been. Yeah. And if you need any extra water, you can use the ones there. It'll make it easier.
You want to get on? Five. Yeah, what's it now? Exactly. No, give much. I'll just give her this.
If anyone's having difficulty accessing this, let me know the Excel file. All groups have shown me their values. Are you guys done? Yeah, can you show them to me? Or you guys are separate, right? Or together? The same group. Okay, perfect. Okay. Any other groups? I have 2 groups have shared with me their data. I'm waiting for 4 more. I know one is still working on it, but let me know other groups what your results are. Control. So but you're the control, right? Okay. 188. Is that what you said? 92 for B. Okay. I appreciate it. Your group gave me 0.625, right? The value for 0.625. Oh, 1.25? They should be there. They be there. Okay, keep it up. I don't want to distract you guys. I'll have a look. Okay. I'm just gonna ask folks, if your group is done, please let me know. You can look at exercise folks, let's pay attention. You guys can look at exercise 2 for the mark recapture method shown on page 78 of the lab manual for our final exercise, one of our final exercises. Page 78, once again. For cleanup, use the to rinse off your lemna into the bean. Please be mindful you don't drop the lemna into the wrong. Any other questions? Okay, I'll talk about this now. Folks, so actually, let me stay on track. I have it reserved for the last portion of the practical, to talk about it. But let me follow up with everyone so we're on pace for time. How many of you are almost done? Because I see that there's 3 more columns left. I know 2 groups are doing it here. Is one of the groups almost done on this side? Jaden, are you almost done? Okay. Wasi, is your group, Nabiha, you guys are all done? Okay. If you can show me your results, I can upload them accordingly. Okay, that's okay. 1.25. Sorry, continue.
So the first one was, I believe, 284.
4,
Next one was 160,
16,
244.
244,
And then one was 375 and 357. And then we're just waiting on the last
and 387,
357.
357. Are they all going in the order like A, B, C, and B?
No, sorry, yes, that's it.
And for which conditions? Is it 1.25 and 2.5? Or I want to know which ones.
Let me ask.
That's
Actually,
okay.
uh, 2.5 for the last 2 values. And then for the first 3, they were the control.
I don't think they're the control
How so?
because they already have values.
I don't really. Okay, then I guess we were the one. Oh, actually, no, like, yeah, 1.25, actually, I'm pretty sure. Because I remember seeing, okay,
Let me,
look in my mind, I think.
it's okay. It's okay. Can I confirm though 270, 375, and 357?
Yes.
Let me have a look as well. I'll have a look at how you guys have done.
Member from Philippines group can help out with section. We really appreciate it. We need one more hand on that. 1 or 2 maybe. Oh, you're the team. Okay, that's okay. That's okay. I'll actually say it.
Well, but it's in the, 1.25.
Yes.
Yeah.
Jazakumullahu. Oh yeah. Yeah. Jaden, when you're done, let me know. Jaden, when you're done, are you almost done? Okay.
It's C 2.5 grams per liter and it's 555.
55, okay. Not, well, it is for now. Great job. I'm gonna say two things. One is cleaning up. You're gonna open up, sorry, you're gonna bring your plate, essentially rinse them off in the basin. So I'll show you. Before I do. Folks, I just want to make sure if you notice there's a bit of variability in the result. So I really do hope you guys have been keeping the track of numbers of value because you can see for some groups there is either a big drop or a big peak upwards. So just bear that in mind. I want to make sure that these values are indeed reliable because otherwise they may obscure your reading. So just keep that in mind. Folks, I recommend just making sure that you find out others that may need help on the other side. And that way we could ensure that they are able to, all of us, complete the task. Don't start exercise 4 or exercise 2 as of yet. I want us to do it all together. I'm gonna give you 7 more minutes to complete the remaining amounts. Try to be as efficiently as you can.
All right, what's your minus 300? What's that? 1,002. Yeah, 350.
So, okay, give me, what was that one? Your first rep?
No, this is B.
Okay.
So 1,000.
Okay, just for context though.
What's the 300? What's the 300? What's the 300? It's okay, they didn't teach it from me. Give us 1.1.
Okay, interesting. For which rep is that? Okay, 3. And they're doing rep C.
Can you guys count them one more time? Yes, yes. You're joking. I'm not joking. Now, at least one more. You want to know why? That's it. How are you doing? Okay, it's okay, just count one please for validation. The reason I'm saying this is because I'm noticing some of you are getting overestimated values. For example, if I see this plate, I'm gonna guess maybe 70 or 80 answer here. If I do the math and I divide it accordingly, like so let's say 80 and 40, right? And she has 3 plates. I'll be like 17, right? But I'll be part of the all of them. Because I can create with them and then they look like that. Yeah. Because we come. So it's 2. That means it comes to be down with well, the reason I asked this was because there is a range, and I'm just a bit surprised how much of it. You got 200-ish, right? 285. But that's already at 900 or something. And whatever. I hope. Should have signed I think I'm all the place. This is Because it was about 1,000.
So, okay, just to confirm, 1167? Okay. You can rinse off, so this was a bit fun, as you can imagine. I must say, I was expecting values a bit lower, so that's why I was interested to know your values. Another pointer though, I'm curious if your values to start off with were more than 12. Folks, let's pay attention. Are we all done? The exercise, yeah? Okay, so let me ask this follow-up question. As you can see on column D, there's a wide variety of numbers, right? Some going to the thousands and some in the hundreds. And what I'm noticing is that, as you can see, starting off, the values are very high. And my concern is that the initial number of thalli were larger than 12 or greater than 12. So I just want to ask, confirming with group, the control group, if you recall from your leaves from the past, if we start off with just 12 or maybe 13 or more. 12, 100% for each graph? Yes. Yeah. Jeffrey as well. Can you confirm that? Yeah. How many? Because I know it may sound like that is, but the issue is that this growth rate is going to be very large and it may be incomparable to the other results. Now, if you compare essentially with, and I'll ask you the last confirm, but the idea is that if you compare these other values, you'll notice what you should expect is that it should be a decrease, right? In terms of the number of value reflecting the growth. What we're seeing though is that if you just do the means, right? Because the starting value numbers should be consistent. Once again, if you don't have consistency, please let me know so I can update these. But if they were assumed to be consistent and controlled for, then you should see a decrease over concentration, or increase in concentrations of NaCl. However, it looks like it's actually going up in this region. So this actually lets me ask, yes.
Actually, can I be 357?
226 for 2.5?
Yeah.
Okay, 276?
226.
226? No problem. And the other plates, can I see those? I actually would prefer if group with 5 grams per liter can update, or not update, but like review their numbers accordingly.
What's the 260?
260. And then 357. What's the 357?
The 357 one was 226.
Okay, so what was the 375?
375.
But you guys want to make sure you're reliably taking count once again, because the numbers can actually affect our readings, our results as an entire lab. So please bear that in mind. I want to make sure that you guys, see and actually can relate back and reflect back to what your results truly depict. So bear that in mind. It's okay. It's okay. I'm just thinking what we should do. Let's keep the 326. I think it was 326 or 375. Okay, so don't worry, you guys, in terms of your results. Let's see. How about for 5 grams per liter? Do you still have those leaves or no? No? Okay. But are you 100% confident with your results? This is why, folks, please bear in mind that you're counting reliably. And I'm not, in no way am I pointing fingers. I just want us to realize that, sometimes if you overcount, right? Let's say you skip a number or you skip multiple numbers, that changes drastically over time the number of thalli that you see, right?
I get it. But like 1, 2, 3, 4, 6, 8 people commented.
You guys counted them separately then, right? So the other thing is you want to bear in mind. So this, like in psychology, you have inter-rater variability, right? You have like essentially people counting in different ways, and maybe not always following to the T what I've mentioned before. But remember, like if I count Dahlia's folks, let's say this is my plate right here. I grab with an inoculating loop one lemna. I see it say or show 4 leaves. I place that down, and then that's 4. Once I grab another lemna, I see another and it says 3 or it shows 3 thalia. In my entire plate, I should have 7. Keep counting in that way. Don't skip a beat because you skip a beat every time or multiple times. That's where we're getting over over 1,000. So just and by the way, it is possible, but I'm just worried that it may not be like the jump is too large actually. But that being said, it is possible NaCl could have immediate drastic effects on lemna growth. But nonetheless, we'll record what we have. And the reason I say to validate this is so that way we don't run the risk of, seeing results that actually didn't occur. And so hopefully the results that we have here are reliable. Yes. Are you sure though? Yeah. I mean, like actually, let me go back because it's 100 gap, right? I don't want us to like clear. I see. Also, okay. Um, I'll just go with this just to be conservative because the other one that we had was 375, just in case. Actually, okay. Even though it may be appearing a bit more larger, I just want to make sure that it's reflective of in case any additional plants were missing to account for them accordingly. Let me see. I may actually adjust something. Um, Give me like a minute or two, folks. Okay, so let me just address as well, folks. Please make sure that you have lab coats that are buttoned up, right? We're still in a lab, so just bear that in mind. Button up, please, your lab coats. Okay, so we'll leave the data as it is, or if I make adjustments, you'll see them. I just want to make sure, folks, let's pay attention because you also have this as part of your lab assignment, or at least what you need to complete for the practical. Bear in mind, you have a formula here, right? So what you're going to do, I recommend essentially going on Excel. You can download the sheet and use a formula. So for example, as you can see, the formula is shown here. Ln N2, which would be this number, brackets. Yeah, I would recommend putting brackets. Ln 2 minus N1. And then you could put larger brackets outside. Oh. You can also use square brackets, I believe. That should be okay. Not just in case. You do this divided by how many days? 14, right? So, and I want you to bear in mind, folks, please let me know if you did not have starting 12 leaves for your thalia, because this has a great impact on the results that you would see. And essentially, you could replicate this across all your cells, so you don't have to repeat it. They'll show it all around. And then your next step is essentially taking the mean, right? So many of us should hopefully know how to do a mean. You could do the geo-mean, you could do average. Average is also reliable. And then so you take the average of the 3 here, and then you repeat it for the other ones. When it comes to your percentage growth, actually, I want you to realize that this technically is your control, right? So bear in mind that this should be 100%. Because your values are going to be compared to your control. And this is why, folks, you need to be very mindful of your starting and ending values. Once again, if your ending values aren't as they actually should be, then you may actually see variability in your results across concentrations of NaCl. So, like, for example, I'm going to do this for okay, actually, let me just show you the mean for 2 more. For those that are also wondering, you do average or mean. I'll do it just for 1 more, actually. Geo mean should be also okay. And remember, you're doing it just for the 3 each rep, but be consistent in the formula that you're doing. I'm just showing it for simplicity. If you want to get the percentage, what you do is divide by the mean growth rate by the control. So the growth rate, the mean growth rate of your experimental condition by the mean growth rate of your control. And you can do that, multiply by 100 or 100%, and you should get your results. Here it's not shown as percentage form, but you can convert it, and you should be able to see either here or on Excel on the app, like the desktop edition or version. You should also see an option directly to convert it into percentage form. So bear that in mind. You're doing this across all of the ones shown here, and then you're gonna not only tabulate them, I have either a table, but likely have a graph inputted for your assignment. So just bear that in mind. If you guys need help, or need me to clarify this, let me know. But yes, I'll be happy to answer your question. Whoa, oh, I have to go to random files. So sorry. Whoa. There's something I have to show you guys. Sorry. Your question. Yes. T is 14. 14 days. Folks, so great question. Anyone else have questions? Because you guys need to know this, by the way. And you obviously want to get reliable marks, because if you, for example, don't do this accurately, and let's say don't inform me that you've changed the values for your leaves, then you're going to see values that not only impact the control, but also the other values, the other experimental conditions. So please bear that in mind as you want to be consistent or accurate in the results that you have. Any other questions? For control? Control is set to 100. So it's already assumed to be 100 because so the control growth rate is the growth rate of your control. And because you're comparing the control to the control, it's pretty much 100. All the other ones, so even though it says, let me just update this so you understand what it's referring to. Percentage growth rate, folks, let's pay attention. Relative to control. We can make this a bit larger, but the idea is that, and this is what you want to be able to understand logically and also for your data analysis, because your control reflects the growth rate or impacts, relatively speaking, what your further analysis, or essentially you want to use your experimental results, the mean values that you've gained from growth rates, to compare them back to the control. And that's referred to for your percentile growth rates, shown in column G. Yes. Yeah, yes. Well, so, sorry, you want to say it one more time? Great question. So, okay, let me clarify this. Folks, we're still in the lab, right? So, okay, let's answer Jane's question. This is a very important point. As you can see, there are exponential growths in the number of thalia in total for Lemna's plants, right? You had only 4 starting off, but as you can imagine, you have many more, right? Many more Lemnas in each container or each cup, and each of them having either the same or relatively more number of thalia or thalli. So bear that in mind. The values increase, right? As we saw from the control, they went to the thousands. But the growth rate shown here is in ln. So remember, this is a logarithmic relationship, meaning that even though it's like in decimal form, like in less than 1, that reflects a large, and by the way, this is every day, per day. So even though you're dividing this by 14, and that's where you're getting it less than 1. Imagine if you multiplied it back by 14, your values would be at least in ln 3, which is actually a great exponential increase, and it reflects pretty much the 1,000-fold increase that we've seen from your results shown here. But to reflect this, your mean values, your growth rate reflects the values in the formula in ln. So bear that in mind. You use the ln for your formula, for your calculation, and then your mean reflects the average of all 3 reps. And then you're doing the percentile or percentage growth rate relative to the control. So if I, let's say, did this one once again, you get the mean, right? So you do this formula. I did the formula shown here. You could do it across, press enter, and then do the average. I've done geo-mean, but do average. That's, for many of you, maybe more easier to understand. And then you should get a very similar value. And then essentially what you do for getting the percentage is grabbing the mean growth rate shown here in ln for experimental condition divided by the control's mean growth rate in ln, and then multiply that by 100%, or just convert it to 100%, convert it into percentage form, and then you should get your values accordingly. All of our values should be under 100%, so bear that in mind. If you're getting 1,000% or 172%, then likely something's wrong. Just bear in mind that you're able to understand how you would do the same for the other values.
You take for control, you take the control with, and now you take the lead from that and just like.
Folks, I can hear people speaking. Please bear in mind that that's disrespectful. We need to pay attention. Continue, Jaden. Sorry.
Control it, we control it, and then just about experiments to get it right on.
So you would actually do the other way. You would divide this value by this value. Oh, divide the experimental by the control. Simone, are we paying attention, folks? Okay, so I hope you guys understand this. Very, very important. Did I answer your question? Yes. I will just post it with the first two. This one you guys could look at. Actually, I would prefer if you guys do it for the others as well. Technically, I wasn't supposed to. I just wanted to show it to you guys, but you guys could do the exact same, and it'll be easy to do. Trust me, because it's not too difficult. It's very simple. And this may also help you for understanding the other values. Yes. Pretty much you slide it actually. So you see that small bottom right side big square, that's square. Yeah, and then it drags and then it goes to all of them. I recommend doing on the desktop if you can. Now, folks, I want us to actually go straight to, because we're tight on time, so let me go straight to the remainder of the practical. Exercise 3, you could see on Quercus. Populations. Now, this is one of our final exercises. I want you to have a look at your apparatus. You have blue containers on each bench, right? And pretty much you're going to be looking at the population of beans. So you have beans in a small blue container, a blue vial, like a cylinder, and you have rice in the larger blue canister. And the idea is that, like individuals shown here, populations live in a particular location, and ecological communities make up multiple populations of different organisms. So it could be even you in a lab. You have bacteria, we have lemnas, we have daphnia, right? So think about integrating our past, projects or exercises and concepts together. Even like being at the zoo, it's a community, right? Filled with different organisms from different pavilions. And so this is something that we're looking at in another way, comparing different results and different values. While we're not accounting for some stats shown here, some of this is important for you to understand. For example, the effects of population size, the parameters of birth, death, emigration on population size, right? To going inwards, to leaving a population. And essentially, this is something that ecologists look at in real time using graphical analysis that we've done or that we may be able to do from the lemnas in a reverse order. But nonetheless, that's one method or one concept to understand. Any questions, by the way? I know it may be a bit quick, but let me know. Folks, please once again pay attention, because I do notice those that aren't paying attention, even if they don't think I can't see them. So just bear that in mind, please, because you want to do your best, and I want all of you to perform well into this course, but you have to also put the effort that is deserved to ensure that you deserve the marks that you get from the lab. So just bear that in mind. Now, in terms of population size, right? This is shown in log as well. You'll notice the functions in E. So this is actually referring back to our results. hypothetically, if we didn't just do like road salts, but we use a stimulus, let's say like if you recall from our previous exercises, certain nutrients or certain, like even in AO1, macronutrients, micronutrients that would, like for the winter wheat, boost up certain growth patterns. The idea is that you could actually see things going up over time. But then you have like conservation efforts that we talked about at the zoo. So you want to bear in mind some populations may go down over time, like the winter wheat, following drought conditions. So you have different, patterns, traits that are exhibited over time. And so this is a way for biologists or even just people of awareness to understand or to be able to perform greater analysis of populations and communities around the world. Now, for your exercise today, you'll be doing mark recapture of looking at beans. So you're going to be labeling the beans. I know you see, a red cardinal here, like the zoo. But essentially, the idea is that you're going to follow mark recapture following page 78 of the lab manual. What you need to do is the following. This slide should suffice. Let me go to this page 13, slide 13. I want you guys to look into your manual. You can start off with page 78, but you really need to bear in mind that you're paying attention, right? So this is once again, when you're final labs here at UTSC, please make sure that you're paying attention, performing to your fullest, right? So, because it's not just about me watching, right? There's someone far more greater than it. So let me show you what you guys can do. You guys can open up your ASIC build of rice. Make sure once again that you're not like thinking about with the rice open up in the air. Right? You want to return all the rice back to the container. Then as you should have a small 50 ml funnel of two beans. You're going to open up those beans. And you're actually place them inside into your box. Actually, then all of you guys that was every question would efficiently as possible. So we should close, by the way, the whole list closed. Then these are inside, rights inside, all the things are here. So it's a kind of bit of fun. What we've done, you open it up. And what you're going to do is reach your body where you're up to the one which is again, or you find it already good at this. You actually need to you're going to do the following. Reach your body all the touch that you have, or hold it at the bottom, the holes, for rice or food come out. You're going to just take the hole, you're going to go inside, over to the bottom. And then you're going to use one of these pesky plastic holders to essentially go inside, all the way till you press into that into the cup inside. And then because the idea is that you already have rice and beans inside this cup, and so by pressing inwards, you're going to remove any extra straight into the container. Right? So just try to keep the rice here. And then when you're done, it's inside here. Start counting your rice. Counting your beans. And you're going to be counting the beans. And as you can see on the slide, I want you to bear in mind that you follow the instructions here. Because you don't want to error in your exercise. You're going to count the number of beans and essentially continue this process. So count the number. Let's say you have 5. If you have 1 exercise, you're
only one. Okay, so you want to actually keep going until you get 20. So some of you may get like 5 in a cup, 4 in a cup, just keep going until you get 20. That's my recommendation. And then when you get 20, label them and follow the next steps in the exercise. We're going to talk about that in this scene. Actually, another idea. Actually, let me know how you guys do for the first 2 counts. If you find less than 10, let me know, and then we'll just make some adjustments. Any questions, though?
After we like just have like never concept, put it back in, take it back out.
No, there are student steps where you are, student steps where you aren't. So let's bear that in mind. They're in the manual, but they should also be on the slides. All credit for you guys as you go through it. Let's start off with marking 20. You'll mark them really shortly. Try to do it on the surface of the beam, and that way you can be able to differentiate them accordingly. Also, please hold budget timing. We are not going to time. I want to make sure that you guys do. Final question. You guys can go straight ahead and let me know how your counts are for the first
20 beans, right?
Okay, let me no.
Okay.
What you can do is mark it.
Yeah, mark them.
Mark them and
Okay. And then we'll put it on the street line. They will stop. Or do I put the wall of carries of beans?
oh man.
So this time we
I would recommend
in the middle.
keep doing it in the center.
And then that wall like something and remove it, and shape it and put it on the right side.
Your steps where you're gonna take back the beans, and then don't put them all the way back in. You actually that okay, Jim? Yeah. Okay, so folks, let me just ask how folks, we're going to do a brief adjustment, and what I want you to do is just grab 20 beans, mark them fully or at least halfway. So let's say, I noticed some of you may just make a single streak on your bean. Try to make it cover up so that way it'll be easier to discern, right? You can make a larger streak on the surface of your beans, but do this for 20 of them and then place them into the container. Then mix them. And then I want you to start doing those cups, like counting the numbers that you grab. And because the idea is that you're marking them, right? Mark and then recapture by seeing the number of beans that remain marked and those that are unmarked. And that you're going to be counting them in your table that is shown, like you're going to record all this data in page 82. So I recommend doing this now. And the idea is that we'll repeat this for until you get 20 beans in total. Sorry, yes. Yes, so mark 20 beans. Just grab the 20 beans. Just mark them, right? It should be easier to do now. And then you just mark them. Try to cover the surface so it's distinguishable, that you know these are beans that you marked, right? And then like on both sides, ideally, or every side that like every dimension possible. And then be able to place all of the beads, all of the ones that you have from that vial into the container, mix it up, and then you should, inshallah, let me know what your values are. Place them into page 82 of the table in column, the column indicating the first experiment. That will show you 20 marked beans, but you want to showcase to me, based on 20 captured beans, how many of them are marked in total, and how many of them are recaptured. Any questions? Yes. It's okay.
So like basically marketing is the magnets and then try to get rice that's unmarked and try to get beans that's unmarked, right?
Yes. Yes. I'm gonna draw it out. Or let me just say it one more time. No, it's okay. It's okay. I'm gonna say it one more time, folks. I want to make sure it's aware because it is an adjustment into the or something to keep in mind to make it more efficient. Your first experiment. We have 3 experiments, mini experiments in this exercise. But the idea is that for the first one, you mark 20 beans. Just take out 20, count them up, mark them accordingly, and then place all beans. There's more than 70 ideally. So you'd have like a good number of them in your into your container. Mix them up. Use the cups once again. Use the cups to go down into the bottom of your container, into the center, I recommend, of your container. And then use the, yep, good job. You can also use the one there. That makes it easier. And then see how Jai is doing. Essentially, you place the plexi or like the thin film paper or plastic underneath the cup, so that way it easily slides through and you're able to lift it up with the remaining rice and beans. When you're done, essentially place back the beans, record, count the number of beans that you have. They should slowly go up to 20. And inshallah, the goal is after that, you're gonna record those values. I'm gonna lift this line up. Yes. No problem. No problem. So hopefully this table makes sense for you guys. So mark 20 of them at the start, and then place all these inside. Then mix it up. Start putting your cups inside. Place them separately in the container. How many beans that you have? Try to go up to 20. This is what you see for factor over 2. So keep going up to 20. 20 total beans, right? So once you reach 20 or even 18, that should be okay. And 20 is ideal. And then start recording those values in your table on page 22 for the first experiment. You'll record a mark, which you have with 20 roughly, right? Then C is the number that you totally recaptured from the number of times you've been using the cup to recapture. That's the recapture number for C. The number. Oh, I think there's a small edit that needs to be made. Yes, total recaptured is C, and then total recaptured that are marked is R. That's okay. Yes. Just keep counting, keep rounding up until you get to the rest. Try to put back. I recommend putting it back each time. Just keep going up until you get 20. But essentially use the cups to count and to count the number of beans that you have and the number of remarks. Done exercise one, i.e. we've grabbed 20 beans in total. Have all of us grabbed both 20 beans, just to confirm once again? Yeah? Okay. So, let's see. Our next step is exercise two. Or experiment two, rather. And I want us to focus now. Folks, we're all done exercise one. Almost? Okay, I'll wait. Let's wait. By the way, the two of you, Simone and Nia, can you see the slides or no? Yeah, okay. Okay. So now that you've had hopefully 20 captured beans, remember that even if it's like, so you have 20 now from 20 that were initially marked. M refers to marked beans, so that's 20 roughly, and then you have 20 randomly captured or captured beans from the cups themselves, right? So that's exercise one. You want to record essentially the number of beans that you've recaptured. That's C in your table on page 82, and R is the number of recaptured marked beans. So bear that in mind. look at the beans out of the 20 that have been captured now. How many of them have the mark of the Sharpie on them? And that's what you want to record. Now for the second experiment, remember, you should have the beans still out. Take back the rice inside into the experiments, but keep the remaining beads, the 20 beans, outside of the container. The rice, because it's part of the experiment, right, sample, you want to be able to return it back into the container each time, but leave out the beans. So you have 20 beans that are left out, right? Some are labeled and some aren't. Your next step, let's have a look at page 82 and page 81 as well. You're going to continue recapturing another 20 beans, right? So you're going to continue this process, capture another 20 beans from your cups. So keep capturing, and once you're done those 20 beans, count all of them together. Actually, not even all of them together. Count. Yes, actually, sorry. Count all of them together. Count from 40 now, from the 40 beans, right? So think about it like this, folks. You're changing the number that you've captured. You still have those 20 beans that are out for exercise 1 or part 1 of this final exercise, exercise 2 of the lab 6. The idea is that for experiment 1, you've taken out 20 beans. Some are marked, some aren't. Now you're going to repeat that process, leave those 20 beans there, but repeat the process of using the cups to grab another 20 beans. So recapture, again, another 20, and then count the number that you have now in total. For the number of recaptured beans, which should be 40, and then the number of marked, which is generally 20, and then importantly, the number that are marked and recaptured, which is R at the third row. C will be the total, but R is the number of beans that are marked. Yes. Folks, can I just get an update? How many of you are done exercise 1 or experiment 1? How many of you are done experiment 2? Jai and Nia, are you guys done experiment 1 and 2, or are you into experiment 2 now? Into 2? Into 2? 1 or 2? Okay, so continue going. We'll wait another 2-ish minutes to help others record their data. I want to make sure that we're all consistent. And let me know if you're having.
thereby, how did you just one, really not one market, just a second. If you didn't know what it was. What was first and I want to make sure you guys understand this last part before we So now we experience. So before doing so, so folks, for just to explain for exercise 2, by the way, technically it's okay to keep the marked as 20 because I know you so marked in the system in the sense that in the environment they would technically be 20 that you would have had right initially marked. So keep it as 20 for now. Actually I want to see because we had another setup. Yeah, leave it as let me see. Data. So leave it as is. Leave M2 should be also around 20. Now the question for your third experiment, you are marking 20 other, so remember you had 20 marked, right?
from experiment 1 and 2 in total. Now your goal is to get 20 other unmarked beans and mark them up. So that way you should have 40 marked beans in total. For exercise 3, you're marking another 20 new beans, 20 unmarked beans essentially. So that's where you're getting 40 in total. And now you're gonna essentially, yes. So keep them filled into you're gonna put all of them together. So
All of them are equal to
all of them. So remember, okay, let's say you had 17 marked beans following experiment 1 and experiment 2, or even just experiment 1. If you had 17 that are marked, for exercise 3, mark another 23 beans that aren't marked, essentially. And that will make 40. 17 plus 23 is 40, right? So you're gonna have 40 marked beans in total. Now your next step, folks, is to essentially repeat the process of using the cups to capture 40 beans in total. That will include beans that are marked and unmarked. And hopefully, inshallah, you should be able to complete the remaining third experiment. For M, the number of total marked, which should be around 40. C, the number of total recaptured, i.e. roughly also 40. And then the one that's really interesting is the number of marked and recaptured, or marked recaptured beans, which are beans that you've recaptured but are also marked from the 40 that exist. Yes. Sorry? All the bricks back as well. If you don't want to do it on the floor. And also, I recommend, even as you begin shaking, picture all the bricks inside. That way, it's like, part of your model. Folks, so I want to just clarify this one more time. Please make sure that you have all rice back into the containers with the beans. There should be now 40 marked beans. Remember, 20 from the first experiment, 20 in the last. And now you're actually going to connect things really nicely as we saw from our past experiments. Your next step is pretty much repeating the process of capturing 40 beans in total. And inshallah, you're going to now see your last value, R, and what that indicates for the number of marked beans from your 40 that you recaptured. So bear that in mind. M, C, and R for experiments 1 to 3 should all be labeled in your table. Page 2.
I'm going to try to
You guys, if you have any plates, please let us know. Please bring them back to their location. Make sure the Lemna are also here, and then essentially dispose of the pond water. When your group is done and has calculated or different M, C, and R, let me know, and we can quickly go over the final slide. They're free guys. Robert, If you guys want, folks. So I just want to make sure that we're all on the same page. Once you're done counting M, C, and R, respectively, M is the number of marked, which for exercise 3 should be 40. For total recaptured, that's C. And then R is the number of marked recaptured beans. So from the ones that you captured, we should also be 40 for our last experiment. How many of them you have are marked? So try to derive those values. When you're done, this is what you need to do. Please bear this in mind. You don't have to, okay, to make it easier, when your group is done, exercise 3. Folks, when your group is done, exercise 3, bring your rice all the way here and place all the rice and beans in here. The beans should come up to the front, out to the top, and that's all. And the idea is that you want to count the number of beans that you have in total, right? So if you haven't counted the number of beans you have in total from your tube, you're going to return them back afterwards, but use this to help essentially filter out in the basin your beans from your rice, and you're going to put the rice back into your containers thereafter. You could count them also here, it'll make it easier, and then I'll place them back into the tube, the little sonical 50 ml tube here, and then into your blue container with the remaining rice. Please make sure you don't spill, right, onto the floor or anywhere. You could use this tray once again to place the rice, and then place them all back in here. Any final questions? And I will be, that should be most of, but I'll be more of the handy with the remainder of the time. Just bear in mind if the beans don't come out through the basin, sometimes they may be small. So if they are, you just want to account for them in your final count for N. Just back to the one container. By the way, once you're done, you could get seated. And there's a bit more to complete before you go leave. Before the end of the
With all the genes that you counted as the final counter, those is N, and that's one thing is true. With N, C, and R, a positive term in the front of it. I recommend actually at the very end, try to put the rates essentially back.
No, I know, but I'm making this. This time of the day. No, take your time. Nice, it's not fine at all.
By the way, that all these are restricted back to the
Still in utility. I know, I'll get that.
So please, once again, try to organize, conventional,
So, those who want to count. Remember, you count your final number of beans. Please do so, as that's part of the table at the very end. So make sure that you count them all, and inshallah, that should be okay. Then, when you're done, let me know, and I'll try to see our next step for the end of the day. No, it should be for n should be lowercase n should be a your total number of beans should be around 75. Like, sorry, can be 70 to 80. But make sure you have the precise number for comparison with what you expect from what you've observed. Yeah, you need to count the beans. Oh,
So that is the answer.
no, that's.
Oh, no, and. Someone may have well, moment. Let's go. Get things sorted. Okay. So hopefully all your benchtops are clean, right? For the upcoming practicals, I see that there's 2 or 3 plates that are missing on this side. If you have extra plates, please bring them this way. Nia as well. Simone, please make sure you grab any extra plates. Yeah, more than 3. Place them back to where you found them, and we'll continue to end. Yeah. Place them in the place where they were before, because they were consistently either large or small plates. Try not to have extra plates that weren't present. But let me just go quickly forward to. I know. Yeah. Okay. That's okay. So let me just briefly highlight. Folks,
you have the results, you have the calculations, or sorry, the formulas in the lab manual for you to be able to derive the next sets of data, and you'll see it on pages 79 and 80. For the lab 6 assignment, you want to make sure that you have this figure made, and that will incorporate what you've expected, right, from your calculations of n, the capital N, based on the formula shown on page 79. And inshallah, accordingly, comparing that with n lowercase actual population size. So you want to be able to have those values accordingly with the error bars that demonstrate your variance. And that's actually highlighted as the confidence intervals. So essentially how confident you are that your mean represents the actual correct number of beings. So bear that in mind. And then we've already done cleanup. Make sure you have a detailed figure caption. Once again, figure caption below. The lemnas we've cleaned out. Let's see. Plates, once again, make sure that they're in the right location from where they were from before. Please make sure that you do this as well. Answer all questions, right? Now, briefly, I just want to highlight 2 other things, or maybe 3, in that, so if you're on Quarkus and you wanted to see exercise 3, as I briefly showed, actually, let's go to your manual so you see it more directly. On page 89. So you will have the data already presented on Quarkus, and your goal is to actually plot that data so that way you can complete it after the lab, and that's okay, right? To do for timing. That's exercise 3. We've done exercise 4. We've done exercise 2 and 1 with your pre-lab. Exercise 5 is your assignment, which is also due 24 hours. But make sure that you are able to answer the details that are shown in the manual, but also from what's replicated online. So, for example, the mark-recapture method, there's specific questions that are asked based on your results. And from the Lemna experiments from exercise 2, you should be able to describe what you've seen on Quarkus. And that's the separate file. There are 2 separate files. One's exercise 3 and one's exercise 2 of what we've done about the road salt and experiments of Lemna growth. So that pretty much concludes, or that almost ends off, what we have to discuss. There are, or briefly as well, make sure that you describe your results, interpret them, and provide specific details as to whether or not they complement your expectations, following the steps and questions in the lab manual. And you can find them as well on Quarkus. I'm going to talk about FLR now. I know some of you have seen your grades and maybe are wondering how was I graded. I grade, so like there are many. Overall, I would say the FLRs were good. There are some things that you want to keep in mind, and I'll try to send like a feedback, like a general guideline for those that are wondering, but they should already, you should likely see it also from the Bio Help Desk because they already have an FLR checklist. So if you've used that, then that's amazing. But if you have questions specific for your FLR, let me know in due time. Now, when it comes to your, actually, let me ask as well, because I had specific details. FLR's feedback. So I couldn't give it to everyone as well, feedback, because of the number of students I'm teaching for different courses. So just bear in mind that if you need, if for whatever reason the rubric doesn't align with the expectations or the mark for your grade in a specific section or an accumulation, let me know and I can describe this to you over email. Everyone has my email, by the way. Mohammed.saeed at mail or at utoronto.ca. Both work for me as a staff member. So, and as a former student. Now, before I end off, you likely could see on the slide lab exams coming up soon. I wanted to help prep you guys. So I was actually going to do 2 separate seminars or sessions online. And I actually have dates that I can confirm for you guys if you want. You could take note of them. They'll be done online on Monday, March 31st, 10 a.m. to 12 p.m. for a review of labs 1 to 3. Labs 4 to 6, inshallah, will be done on Thursday next week, inshallah, April 3rd. April 3rd from 3 to 5 p.m. And that way, you guys could at least get ample time to ask me questions in prep for the lab exam, which, as you're aware, includes the majority of the practical discussions, concepts, and skills that we've talked about throughout our 6 weeks together. So, or consider the different questions that you've been able to answer, different calculations you've been able to undertake, and even graphs that you were able to do in the past, and now interpreting those data, values, and results accordingly. So consider these different concepts, the different stats, the different methods, the different conclusions and results, and especially think about the scientific hypotheses like the research hypothesis, alternative, null, etc., and being able to describe details specifically. Very important that you guys put in that effort that's required, and I really do hope the best for all of you. With this, I really hope that you've had an amazing time. Let me know if you have any questions. I look forward to your excellence. I'm done actually. And with that, I look forward to seeing you guys all in the lab exam. Take care. Monday and Thursday, 10 a.m. to 12 p.m. and then 3 p.m. to 5 p.m. No problem. Take care, everyone. Please make sure that you're able to take all your belongings. Keep it up. Amazing work. You guys have done stellar. And I hope all the best for you guys. I have to clean up as well for timing, but let me know if you guys have questions. As I pack, sorry, if you don't mind.
Okay, so for the pairing of pathways.
Oh, I didn't talk about it. Is it okay if I do it outside?
Yeah.
Yeah, is that okay? It will be a Zoom. I'll try to talk about it because I know we're a bit short on time. So I'll try to send, or there'll be an update sent whenever possible. But keep those details that I talked about. Oh, let me know if you have any questions. The feedback form is also on purpose, if you guys are wondering. So I hope you guys will find it of immense benefit, or even just letting me know any thoughts, criticism, compliments, or anything really, anything